So it's a great pleasure today to announce the inaugural winners of the UK Biobank Scientific Impact Awards. So these awards celebrate outstanding research that drives meaningful change in health through innovative use of UK Biobank data. The awards honour individuals and teams whose work exemplifies excellence, innovation, and real-world impact. I'd also like to emphasise the role of the UK Biobank Participant Advisory Group, or so-called PAG, in this, because actually they selected the three winning awardees from a short list of about 10. And they're a fantastically powerful group. They represent, obviously, themselves as participants, but also other participants, and are a fantastic opportunity for us within Biobank to gain information from them and opinion and feedback from them, which is extraordinarily useful, not only to UK Biobank Central, but also to the Ethics and Access Committees. So we're very grateful to them. So with no more ado, I'm going to move on to introduce to you the first of the runner-ups of this new award, Dr. Bingxin Zhao from the University of Pennsylvania, who's going to be online, and he's going to give us his talk entitled Multi-Organ Imaging Genomics to Illuminate Whole Body Biology of Brain Disorders. Bingxin.
Thank you very much, Matt. Can you hear me okay?
Yes. Yes.
Yes. Hello? Sorry. Thank you, Michael, for that. And I'm very sorry I cannot be there in person today due to visa issue. Hello, everyone. My name is Bingxin, and I'm an assistant professor at the University of Pennsylvania. First, I want to sincerely thank the UK Biobank and the committee for the Scientific Impact Award. It's truly an honor. Today, I want to share my work on multi-organ imaging genetics using the UK Biobank imaging study and how this dataset enable us to uncover organ interactions and disease mechanism. So the UK Biobank imaging study began in 2014, and it's truly unique. We now have a brain and body image from the same 100,000 patients. This includes the brain structure MRI, different MRI, function MRI, also cardiac MRI, abdominal MRI, GSA scans, and many more. The scale and scope of the dataset allow us to ask new questions about how different parts of the human body are connected. That will be the focus of today's talk. So our research question here is, how can we integrate the UK Biobank imaging data across organ systems to better understand the disease mechanism and the brain-body connections? There are two motivations. The first motivation, many brain disorders, including the Alzheimer's disease, the AD, are systematic and involving not only the brain, but also body organs. The second motivation is the brain-body connection, which means brain and body disorders are shaped by complex interactions among multiple organs. So our approach here is the imaging genetics, a framework that connects the imaging phenotype with genetic operations to uncover biological pathways, organ interactions, and disease mechanisms. So begin with our first example, the heart-brain connection. So here we analyze 5 brain MRI modalities and 6 cardiac regions. These deep imaging traits allow us to quantify the brain-heart connection at high resolution. What we say is the cardiac MRI traits are broadly associated with brain structure and function. For example, the regional brain volumes, cortical thickness, white matter structure, and functional connectivity. And the strongest connections are found at the brain white matter peak. This illustrates that the heart-brain health are well connected, and the results are discovered and replicated in the large-scale UK Biobank cohort. So here is a more concrete example, but I think due to time constraint, I will just skip it. Really, we found that the greater global wall thickness of the heart is associated with the increased MD in the brain white matter. And beyond phenotypic associations, we try to look at the shared genetic structure. So on the left, this is that showing the statistical colocalization in this gene region between the heart trace and brain structure. And on the right, we further found the genetic connection with brain disorders and cognitive function. So these results illustrate the shared genetic influence connecting cardiovascular traits brain conditions. Now we look at the second example, which focuses on the Alzheimer's disease. A key question to try to ask is how are organ systems and biological pathways involved in AD? And to address this, we integrate the risk gene with multi-organ imaging-derived gene modules from the UK Biobank. And we found that AD-associated genes do not map uniformly across the body. For example, AD module, gene module 1 overlaps exclusively with the brain structure MI module, capturing cortical and subcortical biology. Interestingly, AD gene module 2 overlaps closely with bone imaging type. This kind of specific overlapping helps us decompose AD risk genes and understand AD from a multi-organ perspective. If we want to look at the details here, I will skip due to the time constraint. And we can look at the specific imaging traits across the human body, have very high overlapping gene overlapping rates with AD. Okay, now look at the gene set enrichment analysis to identify biological pathways underlying imaging disease relationship. This provides an organ-specific biological fingerprint of AD and highlights how known and novel pathways are related to both AD across the organs and which are reflected by the imaging data. The top-ranking enriched modules provide a new insight into the genetic imaging AD pathway in both brain and body organs. So finally, we can further look at the results at gene level, and we found that the imaging gene modules frequently have key AD risk genes, and the genes involved in top AD-related biological pathways, as well as active AD drug targets. So these provide a gene-level insight into how imaging traits across organs related to AD mechanism and could help guide drug discoveries in the future. So to summarize, the UK Biobank imaging study is uniquely positioned to serve as a leading resource for organ-specific biology and imaging disease mapping. So we illustrate the power of imaging genetics with two examples. The first example is the brain connection, and the second example is the organ-specific AD biology. So this framework allows us to uncover disease mechanisms across pathways and genes. Altogether, these results will move us towards a more integrated view of human health, and we hope researchers will join us in leveraging the imaging data resources from the UK Biobank to advance the multi-organ understanding of complex disease. And thank you very much for your attention, and feel free to email me if you have any further questions.
Congratulations, Bingxin, and thank you so much for that excellent presentation. We move on now to the second and equal runner-up of this award, and that is Dr. Callum Hunt from the University of Leicester. Callum, would you like to come up to give your presentation? Oh, it's coming that way. Callum, congratulations.
Thank you very much.
You get a certificate with
well.
Sir Rory Collins' signature.
So hi everyone, I'm going to be talking today about some work I've been doing using UK Biobank data to understand more about the development of the most specialized region of the eye called the fovea. So our central vision is our direct and high acuity detailed vision that we use, for example, to recognize faces or read sentences, and it's conferred to us by the specialized pit in the retina called the fovea. And at the cellular level, the fovea looks something like this, where the inner retinal layers move to one side, creating the pit, and there's a dense packing of photoreceptors that enable this high acuity vision. Now, we can actually visualize the fovea in vivo using an imaging modality called optical coherence tomography, or OCT. So you can see on the black and white OCT image the distinctive foveal pit in real time. And this is an important tool clinically to look at a condition called foveal hyperplasia, which is simply when the fovea does not form properly. And for the purposes of time, just to say on this slide that the shared feature across all grades of this condition is that there's a loss or reduction in the foveal pit, and we can see this on the black and white OCT scans. Now, that's what we know about foveal hyperplasia phenotypically, but genotypically we know comparatively less. So most of the disorders which cause foveal hyperplasias are disorders of pigmentation, but more recent association with a broader range of inherited retinal disorders suggests a largely unmapped genetic architecture. And that's what I'm going to talk about today. So I went about answering this question by conducting a genome-wide association study. So this is where we test for the association of genetic variants with a trait or phenotype, so in our case something foveal related. And for this, we require a cohort of individuals with both genetic data and a phenotype, which is where UK Biobank comes in. So our phenotype, we derived from the OCT imaging available within UK Biobank. So I think there are OCT images for 100,000 or so participants now. But actually, in all of us, the fovea differs in its morphology. So what I did is I developed a deep learning segmentation pipeline that identifies key foveal landmarks, and from it quantified foveal pit depth, which is the feature that is shared in all foveal hyperplasia grades. And I did this for over 60,000 participants in the UK Biobank. When I conducted the GWAS, I found that foveal pit depth is a highly polygenic trait. So we found 126 independent signals, and this was discovery led mostly in individuals of European ancestry, but polygenic score analysis determined that these findings were generalizable in both South Asian and African participants as well. But the challenge with any GWAS is to interpret what these signals mean in terms of biology. And I was particularly aware that my trait is one that is developmentally determined. So to meet this challenge, I created a variant to gene strategy of 12 lines of evidence total, which was developmentally aware. So the first set of evidence concerned the annotation of the variants within the genome, so to nearest gene annotation within key gene features, and using the UK Biobank's whole exome sequencing data, I identified candidate genes based on rare variants as well. I also then mapped my GWAS signals using developmental multiomics data from post-conception weeks 8 to 20, which is around about when the human fovea begins to develop, to see in the right developmental context if our signals are regulating gene expression. I then looked at loss of function effects from Mendelian disease genes or mouse ortholog genes, as well as more broadly looking at the regulation of nearby genes with EQTL and PQTL analysis, the latter using UK Biobank's plasma proteomics data. And then finally, one all-encompassing polygenic priority score. After assimilating all of this evidence, I implicated several hundred genes in foveal development, of which about a half could be supported by at least 2 of these lines of evidence, and 64 were completely novel to foveal biology and, in fact, any OCT-derived trait. And what I found is that when I did pathway enrichment analysis of these genes, is that the fovea is patterned by far more than pigmentation. So I identified genes involved in the specification of all retinal cell types, but especially photoreceptors. I identified genes involved in metabolic process, especially those related to retinoic acid, and a number of genes involved in blood vessel, ECM, and cytoskeletal development, which have more mechanical properties relevant for the formation of the depression itself. Now, one interesting aspect of the previous slide, in fact, I'll go back, is that we actually implicated through common signals genes which, when a rare pathogenic mutation is present, cause Mendelian foveal hyperplasia. And this led me to ask the question, are any of the genes we discovered novel causes of Mendelian foveal disease? So I applied this foveal gene list to the Moorfields Eye Hospital dataset, and in at least 4 of the genes we implicated, identified pathogenic mutations which are causative of foveal hyperplasia. So these individuals have systemic disease where foveal hyperplasia was previously overlooked. So in summary, through methodological advancement at the phenotype level and through variant to gene, I've significantly expanded the known spectrum of genes involved in human foveal development. And we identified a continuum of disease where common and rare variants in the same gene are causative of foveal hyperplasia. We've provided a resource, the critical gene list, and the full prioritization framework where these genes can be investigated downstream, for example, in clinical cohorts, as I've shown you today. And I'll end with some acknowledgements and by thanking all the participants of UK Biobank, without which this would not be possible. So thank you very much.
Congratulations, Callum, and thanks again for a wonderful talk. So that brings us to the overall winner of this inaugural UK Biobank Scientific Impact Awards, and that is Ludovica Griffanti from the University of Oxford, who's going to give us a talk on advancing the diagnosis and characterization of NHS memory clinic patients through UK Biobank imaging methodology. Ludovica. Get an award. Yeah. And a certificate as well.
Thank you. Thank you so much to the committee for this award and for the opportunity to present to you today. It is an honor to receive this award as a fantastic endorsement and recognition of the huge amount of work done over the last 10 years by 2 incredible teams that I'm really proud to be part of. The Oxford Brain Health Clinic, led by Claire Mackay and Lola Martos, and the Oxford Centre for Integrative Neuroimaging, in particular the UK Biobank Brain Imaging team, led by Carla Miller and Steve Smith. But there are many more people in the Oxford Brain Health Clinic who worked hard to set up and run the clinic over the years. So many that they don't fit on a slide anymore, and so we have to use rolling credits. But without them, none of this would have been possible, so we want to make sure to thank them all. The overarching goal of this project was to harness the power of UK Biobank, and in particular the brain imaging, to better diagnose and characterize memory clinic patients in the NHS. UK Biobank offers big data acquired on state-of-the-art facilities, but being a population-based study intrinsically so far has had limited access to patients. On the other hand, we have the NHS, where real-world patients sit. But hospitals often have limited access to innovation, and the data, especially brain imaging, is often heterogeneous and unstructured, with most memory clinic patients receiving a head CT rather than an MRI. The Oxford Brain Health Clinic was born to bridge exactly this gap. Patients that are referred to one of our partner memory clinics, instead of getting a CT scan, they come to our center where they get a brain MRI scan with the UK Biobank protocol. And together with this, they also get other enhanced assessments. When they are at the clinic, they are asked whether they are willing to participate in research, and they can opt in to different levels of participation. One of the main reasons why this was feasible was because the UK Biobank brain imaging study optimized the acquisition protocol to be as brief and as high quality as possible, with 6 modalities acquired in 30 minutes. And we've already heard about the great achievements in data collection. What we did at our end was to reorder the sequences, split them, and implement the first 15 minutes as core clinical assessment, reported by a neuroradiologist and forming part of the NHS assessment. If the patient consents to the use of data for research, we get those images for research use. And if they agree to take part in research, they are also asked whether they are willing to stay 20 minutes longer in the scanner, and if so, we complete the rest of the UK Biobank protocol. After starting in 2020, we scanned this year the patient number 500. 90% of people complete the clinical scans, 50% of people complete the research scans, and overall, over 90% of the data are deemed of good quality for research use. We shared our protocol openly, and we produced also educational material on the radiology report that we use in collaboration with Dr. Peter Pretorius. And similar protocols are now being used in several clinics. We demonstrated that the protocol is well tolerated by patients and can be implemented within existing NHS pathways. We think that the flexible consent was key for high research participation, with people choosing to participate in the way that feels comfortable to them. And we also aligned other non-imaging assessments to the UK Biobank. This can be summarized in one sentence: minimize burden, maximize value. With one visit and one set of assessments, we then fill in some structured reports which are sent to the memory clinic. And this has resulted in the clinical appointment with the consultant being reduced from 75 to 45 minutes. And because of the high research consent, we get a highly representative real-world memory clinic dataset aligned with UK Biobank. So what do we do with the data from people who consent to research? Well, also in this case, the UK Biobank imaging study, and in particular Fidel Alfaro Almagro, already worked out how to analyze the data to get the most out of the images in a standardized way. What Grace Gillis did at our end was to adapt this pipeline to, first of all, take into account the fact that memory clinic patients have a higher degree of atrophy and vascular pathology. She added some extra quality control checks, and she also extracted 110 additional IDPs that better reflect the changes that are expected in a dementia population. And many of these adaptations are now informing the next version of the main UK Biobank pipeline. Also in this case, thanks to Grace Gillis and Gaurav Balerao, we shared the code openly to complement the existing main UK Biobank pipeline. And this has already been used by other research groups. Because our scans are reported by a neuroradiologist, we were able to verify that the IDPs that we generate are indeed clinically relevant measures in line with the visual scores, which means that now we can move towards implementing the IDPs and translating them in clinical settings to compare a single brain health clinic patient to the massive reference population that is UK Biobank. Beyond imaging, UK Biobank has a huge amount of non-imaging data, which means that we can look for associations between imaging and non-imaging. But these type of analysis require specific statistical methods that are not directly applicable to smaller samples like ours. So also in this case, Grace worked to adapt these big data analysis to be able to look for similar associations in our sample. And I don't have time to go into the details of the results, but the idea is that we were able to replicate well-established associations that have only been previously found in mostly highly selected research cohorts, while we were able to replicate them in a real-world memory clinic set. And also additional associations emerged, meaning that the combination of the UK Biobank method with our unique sample can enrich our knowledge into dementia. And finally, I would like to highlight a few other initiatives that the Brain Health Clinic was able to contribute over the last 5 years. We are one of the founding members of the Brain Health Coalition, which is a consortium that is working to build consensus on how to design, deliver, and grow brain health clinics in the UK. We were mentioned in the policy report by the Alzheimer's Research UK as a successful case of brain health clinic. Earlier this spring, we ran our first Patients' Day to celebrate our achievements with the patients and with our patient and public involvement contributors. And we've participated and held several public engagement events to raise awareness on brain health, dementia, and the role of brain imaging. And with this, I would like to thank again the committee for the award, leave you with our website if you want to know more. Thank again all the people involved in this study and you for your attention.
Outstanding. Thank you very much indeed. I'd like to congratulate all three of our inaugural award winners this year, and we look forward to more of these in subsequent years. So congratulations to Bingxin, Callum and Ludovica, and I think another round of applause collectively for them would be in order.